Hello everyone, welcome to week 2 of the cell signaling practical. This week we are going to subculture the primary explant we started last week, and we're also going to do a cell count to estimate how many cells we've got growing. So the first thing we need to do is, by eye, have a look at the flask from last week and see whether any of the pieces of embryo have attached to the bottom of the flask. So here's my flask from last week. If we can try and have a look at it. What we're looking for is there's a lot of floating tissue at the bottom there, but some of the tissue, it's hard to see. If you just gently swill it, some of the tissue has actually adhered to the bottom of the flask, which is a good sign that we've got some outgrowth happening. And be careful not to tip the liquid into the neck here, so try and keep the flask as flat as you can. The other thing to note so record in your lab manual the appearance of the embryo, whether it's how much is stuck to the bottom, and also record the color of your growth media as well, because there's a pH indicator in there and you can make some observations about the color and the pH of your solution. Okay, so I'm fairly confident that I've got some cells growing in the bottom of my flask. So what I'm now going to do is to look at my cells under the microscope. Okay, so here's the microscope that most of you will be using today. It's a digital microscope, meaning it's attached to a computer screen over here on the left, and it's got a digital camera which will record the image that we're observing when we put our flask on top of this base here. All right. There's also a lever here which switches the microscope between the digital camera, and if you push this across, the screen here goes black, and now the light path is coming up through here, so I can look down and look at the cells this way. So when you start, use your eyes, not the computer screen, to observe the flask. So get the flask and place it over the light path. Choose one of those pieces of embryo that is attached. So I can see one here that's attached, so I'll just kind of try and line that up roughly. And then I'll just have a quick look. Now, I'm observing at 4 times magnification here. Okay, there's 4, 10, and 40. Start on the low magnification, and then the microscope should be fairly well set up anyway, so I've just got a view here, and I can see straight away I've got a bit of embryo attached to the flask, and I've got, hopefully, I can see lots of small pinprick dot cells kind of growing out from there. Now that I've focused, I'll switch to the 10 times and see a bit more detail. Okay, I'll just focus that. Alright, so that's great. I've got lots of cells. Okay. And now that I've got an image that I'm happy with, I'll quickly switch to the main screen there. And I'll try and bring the camera around, and you can have a look at the screen. So excuse if this is really wobbly. Alright. So I'll just plonk this here. Okay, so hopefully you can see the screen here, and if I just gently move, you can see here's a bit of the embryo culture. So there's a bit of the embryo, and growing out of the embryo here is a big lawn of cells, okay? And if you just sort of have a little look around, you can see the cells. I'll just change the focus a little, so you can see. All right. So basically, you can see there's plenty of cells growing on the floor of this flask. I'll just choose another random area, and there you go. More cells. And you can see these long, skinny cells, which are a dead ringer for our fibroblasts starting to grow. Okay? And I'm in the acquire window here. So there's an acquire image thing here. So if I just click acquire image, I'm on 10 times magnification, so those settings are okay, and I'll acquire that. So that should have now made a copy of the image. Okay. Now if I go to Browse, which is up here, there's a whole bunch of images, and here's the image down here that I've acquired. So you need to record the name of the image that the computer's given it. You need to record the microscope settings, whether you want 4 times or 10 times magnification, and you just need to record as many details as you can, as well as recording the description of the flask and the media that you've already made. Okay, so here are the cells from last week, which I've looked at down the microscope. Now again, I've got a nice clean work area here. I've got some fairly clean gloves, some sterile gloves. The first thing we need to do is to remove all the floating debris, because the cells we want have adhered to the bottom of the flask. Okay, we don't want all that floating embryo tissue. So that can be done fairly easily by tipping the cells, decanting the cells. So I'm just touching everything here, so just get my fingertips nice and clean. So just okay, so that hopefully has got rid of most of the cells that we don't want, the dead cells. A trypsin solution, which we're going to use first to wash the cells through. So first wash 2 mils through. Approximately, just so just gently mix the trypsin across your cells and then discard that. Now, in the trypsin solution, it will digest the proteins that cause the cells to adhere to the flask. So don't leave that first wash for too long, because you'll lose your cells. They'll loosen from the bottom, and then you'll wash them away. So a nice quick wash with the solution, and then I think it says to put a mil in, and then leave it for a few minutes. So. Again, it doesn't have to be accurately a mil, just needs to be an amount. And then red lid. And we'll wait for 5 minutes for that to loosen the cells from the bottom of the flask. Okay, so now we need to wash these cells from the flask into a clean 50 ml one of these. Now it says in the manual to use the DMEM media for this. Okay, so, and it says to wash the trypsin solution into the clean flask, and then to add some DMEM to the flask. What I might do is myself, I might just, hopefully this is the right thing to do, but I might just add the 5 mils of the DMEM to here, and then wash that into the into the other flask. So hopefully it's the right thing to do. Again, you can follow the manual if you like, but it just seems more logical to me to. You might want to use the, be careful you don't spill the solution, but it just seems as we'll have a better chance of getting all the cells if we just and then we'll just watch that. Do one of these. Just washing the cells through. Okay, so hopefully that's everything from there. So there should be our cells now, and we'll just carry on with the procedure. Okay everyone, I've spun my cells, and if you look carefully in here, hopefully you can see there's a small pellet of cells collecting at the bottom there. So it says to decant the cells that are supernated away. So hopefully I'll just decant them away. I'm watching the pellet, and that's fine. The pellet stayed where it should. So there's my collection of, where is it? There's my collection of cells with a little bit of media on top. And I think we add a mil of the growth media. Okay, so some of these cells are now going to go back into incubation for another round of tissue culture. Okay, let's leave that there. So I'm just flicking to resuspend. They resuspend pretty easily because they've only been spun down very lightly at 100 RPM or 200 RPM. Okay, so once they've been resuspended, I can still see a pellet there, so I need to really try and resuspend these cells. There they go. I can see the big clump of cells just popped up. So okay. Now we need to take away 0.2 of a mil to do some counting on the hemocytometer, and with the rest of the cells we will propagate them. So again, just mix the cells. We need a sterile Eppendorf tube for this. And then so again, just help mix the cells just by gently, without being too vigorous, just mix the cells a bit. And then I want about 0.2 of a mil, so I'm just taking to the first mark on the tube here. So there's some cells. Yeah, so 0.1's there, so that's about 0.2. And we'll mix that with trypan blue for counting. Okay, so to the rest of the cells, add and then we'll add that to a new flask. Okay, and that should be fine for incubating for till next week. So hopefully we've got lots of live tissue culture cells starting to grow, and we'll label it up and incubate it for next week. Now, the remainder of these cells you're going to count on a microscope. There's lots of them on your bench tops. You need to add some trypan glue to stain the cells. The live cells will be clear and the dead cells will take up the dye, so you'll be able to do a live dead cell count. Okay, so I'm just working at the bench top now. I've got the 0.2 mils of these cells from earlier. We're just going to count these cells and then discard them. And I also have the trypan blue here. So the first thing to do is to set your Gilson on 100 microliters and add 100 microliters of this. To the cells. And give it a good mix. Okay, here's the hemocytometer, the microscope slide, if you like, that you're using. It's hard to see, but there are some cover slips already in here, so just need to carefully. There we go. There's the cover slip. If you can see that, it's a very fine piece of glass. Here's the hemocytometer. Okay, now there are two counting chambers. There's one here and there's one there. Alright, so if you just take the cover slip, okay, so just lay that in position so it's in place, covering the two counting chambers, and then just pipette the solution at the edge of the cover slip. It'll just take up, it'll just, capillary action just draws it up there. Okay? And again, you can do the other one from here. Okay, so unfortunately I've got an air bubble under one of those slips, but that should be alright. So that's now ready for counting on your benchtop microscopes. I don't have time to go through the counting, just follow the books, it's pretty straightforward. But in terms of the assembly of these hemocytometers, they often cause a bit of confusion. So just remember, place the glass cover slip in place and then add the solution just at the tip there, and it will just get drawn up. Doesn't matter how much solution you add as long as you cover it up, because the excess solution will go into these grooves. So it's pretty easy. Alright, and then you can just do your live dead count from these slides. And then just use the benchtop microscopes here for counting, okay? Alright, thank you.